Okay, so it's my pleasure to have Gabriele here, who's been working for quite a long time now on machine learning for epigenomics. So we heard a little bit about gene expression so far, but we haven't really. Today's kind of the biology day, so we're really going to get into biology a bit more. And one of the most exciting areas, I think, is really these epigenomic assays that they've been around for a while, but we're still discovering lots of new stuff, and really much less is known about the epigenome than the genome, I think, still. So over to Gabriele to tell us about that.
Well, thank you very much for the kind introduction, also for the invitation to be here and the beautiful organization of the summer school. I hope you all had a lovely evening yesterday. Is that working well? I see here a bit of an echo. Okay. You're all very much socially distanced, so despite the mingling yesterday, you're still very much in your own little, but we'll see how that works. So this is a lecture on epigenomics. It's kind of an introduction and a little bit almost like a tutorial, I think, or yeah. So do ask questions if you have any. I'm assuming that there's a very varied background with people from more machine learning, some people probably more biology. So it's very introductory, but do ask questions if you have any. So I'm going to start not to talk about epigenomics, but my first slide is about DNA sequence. I suppose you're all very much familiar with that. So this is a very, very short bit of chromosome X, very, very tiny bit of the genome, of course, and we all know that in our cells there is this in fact 2-meter long molecule that encodes so much information about us. And in fact, if we are thinking about it, it stores 3 billion letters. So it's kind of a proper library of information. And when we compare that with, for example, Charles Darwin's monumentous work, Origin of Species, that's about 4,000 copies of that book. So there's quite a lot of information that is needed to encode something like yourself, right? And of course, it's not stored neatly, this information in a library like that with a systematic overview, but it's quite a mess, to be honest, in the cell nucleus. And we're going to see that in a moment as well. And then on the other hand, we have to think about that you're made up of a huge amount of cells. That's also a very startling fact for me always. So there are about 40 trillion cells in the human body. And the truth is, we will hear a lot about single cell later on today as well, but we don't even know how many different cell types there are in the human body. So that's quite startling. So just if you're thinking about the number of cells, it's quite a large number, and it's much more than there are stars in our galaxy. So it's really a fascinating universe to be studying. What we have to remember as well is that each of these cells contain an identical copy of your DNA. And with that identical copy of the DNA, biology is able to create very different phenotypes, very different types of cells. So I'm showing you here the Purkinje cells, that's a nerve cell, a muscle cell, a T cell, and then this beautiful hair cell in your ear that helps you to listen to what I'm saying and decoding my language, of course. So you see that these cells that all have the same DNA sequence come with very different phenotypes and very, very different functions. So that's quite amazing finding. So also that when you study DNA and mutations and so on, you have to keep that in mind to some extent as well. But then there's something else which is also quite interesting. So this is a human Purkinje cell. But then I'm showing you here another cell which looks very, very similar to that one, but that stems from a mouse. So in this case, of course, we have different DNA sequence underlying these two realizations, but they still present with a very similar phenotype. So what that tells us is that the DNA sequence, of course, is not everything. Rather, what is important is what kind of genetic programs are activated in a given cell. So kind of what books are we actually reading, what programs are we having open in a given cell. And that, so the specific genetic programs that are read and executed to a large extent determine the function and the phenotype of these different cells. Okay, I'm not looking too much in this direction, sorry. Okay, and so epigenetic mechanisms are somehow related to these processes. So which genes are being used, which genetic programs are activated and read and executed in a given cell. And if we are thinking about it from molecular perspectives, there are different mechanisms, different tricks that the cells have in their pocket to accomplish this task. So here on the here you see an electrotomographic visualization of the cell nucleus, and you see the different chromosomes in different colors. And again, it's quite amazing how they are crammed into that small space, the 2-meter-long DNA molecule, and how you can actually efficiently find the particular genetic programs that you are needing. How does the cell do that? Well, there are these main mechanisms now that we are going to talk about a little bit. So DNA methylation, chromatin remodelers, histone modification, and non-coding RNAs. So if you haven't heard about DNA methylation, I'm demonstrating it here. So the DNA strand can be chemically modified by the addition of a methyl group. It's shown here in red. And the way I would visualize that is if you have a woolen thread, for example, you could put a little bit of sellotape around it at certain places, and then you would very easily find this place again and again because your physical properties of that thread have been changed. And so you don't have to read the DNA sequence that is actually encoded in that long woolen thread. You only find the bits that have chemically changed, and it provides a marker. So that's one way of finding, again, particular spots that you're interested in the DNA. On the other hand, so our DNA is wrapped around proteins, and it forms a structure which is very much like pearls on a string. And it's for these structures are called nucleosomes, where the DNA is wrapped around histone proteins. And again, these histone proteins can have different chemical modification, and the modification are then locus-specific, and they can also serve as markers to re-identify particular areas on the genome. Also, the position of these nucleosomes is quite important. The DNA strand that is wrapped around it is always 147 base pairs, and then there's a little bit of linker DNA. And you can imagine that if you change the position of the nucleosome, your DNA becomes more or less accessible. And you can package them very densely such that you can't really access the DNA sequence that is encoded in it, or you can organize it much more loosely, and then the chromatin is actually accessible and transcription can happen, and the genetic code can be read off. So that's happening in our energy depending way with the help of chromatin remodelers. And then there's also non-coding RNAs, which also control these processes, but I'm not going to talk about those today. So these are kind of the three ways that you can create an organization of your chromatin, the efficient storage of your DNA sequence in the nucleus. However, epigenetics has been around for a long time, and there are still a lot of there's still a bit of a debate about what it actually is and what kind of mechanisms are part of what should we actually call epigenetics. So when we are looking back from the biochemistry and stepping back a little bit, so how was epigenetics coined? What is the bigger concept there? And Mark Patashner has, for example, said histone modifications, well, they shouldn't really be called epigenetic mechanisms. So what does he understand with the word epigenetic? Why is there such a confusion around it? And for that, I want to go back again to when epigenetics was first coined. There was a guy actually in Edinburgh called Waddington, and he had this idea of an epigenetic landscape, really, that would help the cells to go from a pluripotent cells through a certain trajectory into differentiated cells which are committed to certain fates. And he was, because a lot of this wasn't known at the time, so he compared that with the epigenetic landscape, where he said it's like a ball traveling downhill, and it has to make certain choices to find its path. And so you are starting from in this analogy, you start from a pluripotent state, and you are moving into the differentiated cell, and you can end up at different positions at the end. So very remarkably, he found that developmental processes are driven by no changes in the DNA sequence itself. So there are other mechanisms that are governing these changes. And so what he was saying is that the mechanisms that govern these changes, he coined as epigenetics. We of course know that the bulk of this work is done by transcription factors. So transcription factors are really important to determine which genes so they bind to a specific binding motifs on the DNA sequence, they recognize them, and they do this bulk of the regulation. And so initially it was also so that the ball can only travel downhill and we can't travel uphill again, but a remarkable remarkable experiments showed that we can actually reverse these processes. And we can do that by only adding 4 transcription factors to differentiated cells to get the cells back into a pluripotent state. So that would suggest that really these kind of epigenetic mechanisms are fully encapsulated in the specific DNA binding regulatory proteins, transcription factors, which can, for example, reprogram fibroblasts into stem cells. So that was the initial definition of epigenetics, but there have been a lot of other definitions along the way. So this would suggest that epigenetic mechanisms are really about control and regulation of gene expression. So I'm thinking somehow, and we have been discussing that yesterday a little bit in the course models, very simplified, but it would suggest that you have epigenetic mechanisms that really drive the expression of genes or the phenotype. And that would mainly include the action of transcription factors. So are the epigenetic mechanisms like DNA methylation, histone modifications, chromatin remodeling, actually epigenetic mechanisms in the Waddington sense? Do they control gene expression? And that's also, so they are highly correlated with gene expression, but in a lot of experiments we have also seen that if we are changing, for example, DNA methylation, overall, the differences that we see in gene expression is not as we expected it, if that was a simple case. So in addition to this model, we can also think that transcription factors could control both histone modifications or other epigenomic modifications and expression, and then the histone modification wouldn't actually be so important. They would be a rather kind of a byproduct of the action of transcription factors. And we can see that also, that we can use transcription factor binding, for example, to predict histone modifications. However, it turns, and then there's a question, why are these modifications actually so important? And there's another aspect about epigenomics, and that is about actually remembering what happened in the cell in a previous state. So another definition of epigenetics was that this encapsulates properties of a cell mediated by the genomic regulators that confer on the cell the ability to remember a past event. So why is that remembering and memory across cell divisions and across generations of cell important? Well, of course, if you're thinking about this picture again, if you want to decide if you want to go at what different places what genetic programs you want to activate next, you have to kind of remember in what trajectory you are. So whether you start in this position or in this position, from here you will not be able to come back to this position, even if you're activating a certain type of program. So you have to actually remember where you have been in the past in order to move forward to the next stage. And also in the long term, you want to create barriers against accidental reprogramming. So once the cell have reached their final destination, you don't want them to change their cell identity. So epigenetics has really a lot to do with dynamic changes during development, for example, but also with stability and with maintenance of a certain cellular state. And as such, they are very heavily associated with cancer, for example. So there was in 2016 a quite comprehensive characterization of cancer driver genes and mutations, and a lot of these guys were found. So these are large-scale genome-wide association studies that looked at mutations on the DNA level that might be associated with cancer. And it turned out that a lot of these mutations are found in genes that are actually coding for proteins that have something to do with the epigenome, with writing the epigenome, reading the epigenome, and controlling it. And that is shown here. So these are different types of tumors, and here are the functional categories for some of the genes that are found, and you can see that a lot of these genes are really implicated in epigenetic mechanisms. So fraction of genes is the color coding here. So you have, for some of these genes, like the chromatin SVSNF complex, it's really implicated in a large number of different cancer types, because it seems to be so important to create this barrier against reprogramming the transcriptome. Okay, so this was kind of the two different sides in the debate about what is epigenomics really. So some said that epigenomic mechanisms really control and regulate gene transcriptions. Others said, no, they don't control it, but they remember when expression has occurred. And then Adrian Bird, he was kind of, I think, quite smart, saying both of these explanations are most likely true in different circumstances, in different locations. So both of these mechanisms are important for epigenetics, to define epigenetics. So, and this is kind of the definition that I'm working with. So epigenetics really is about the structural adaptations of chromosomal regions so as to register, signal, or perpetuate altered activity states. Okay? Questions? So the reason why I kind of banged on about that is that we really want to understand epigenetics in the context of how it brings about phenotypes and how it changes the transcriptome and what kind of function it has. And that is really still quite a mystery and really difficult to do. So we do find a lot of epigenomic patterns that have changed in disease, but are they causal? Are they just byproducts? How important are they, and so on? So that's kind of why I think it is particularly important here to move away from correlation and to think about causation. Okay, so with that, I'm going to go slightly more into detail into different projects that I've been involved in, but also in different, so more broadly, why this is important and why I think you should keep epigenomics in your mind when you're looking at different disease states. So the first project is I'm looking at methylation-dependent gene regulation during neonatal brain development, and very often when we are looking at a project, we also came up with new tools. So there's also always a bit of a methodological part of that. So let's first start with this project. So we are looking at DNA methylation in that case, and we have mouse models where we are interested in understanding how the brain develops just after birth. And sadly, this is actually quite a very interesting project, but the data set is a little bit old due to the fact that the main involved people had altogether 5 pregnancies during that project, which slowed it down, but it created a lot of potential for more advanced computational approaches to study some of the older data sets that are still around. So this is with Sabine, who's now in Vienna, and also Christian Cardona. And it arose from a collaboration with Adrian Bird. Sabine and I were both postdocs in the lab of Adrian Bird together. And Adrian Bird was one of the also pioneers in epigenomics, and he was one of the people who discovered the first what we call an epigenetic reader protein, namely MECP2, methyl CpG binding protein. So I've already said that methylation happens on the DNA, and now you have a protein that very specifically recognizes methylated CGs. And interesting enough as well, it is associated with a very, very complex disease called Rett syndrome, which affects girls. They start out developing normally, but after the age of about 1 year, they show massive symptoms. They lose some of the abilities that they might already have gained. So, for example, if they started to speak, they might lose that ability, or if they started to walk, they might regress on that. And the symptoms are getting worse and worse and worse. And it's really a very severe disease. So it was described as, imagine the symptoms of autism, cerebral palsy, Parkinson's, epilepsy, and anxiety disorders all in one little girl. So it encapsulates a lot of function that we need for normal neural functioning. And so the interesting bit is that this is a very, very complex disease, but we can actually link it to a mutation in one single gene, which is MECP2. And that also just demonstrates to you how important DNA methylation is to brain development. So in contrast to many other neurodevelopmental diseases, which really are associated with a large number of genes now, this is one where we have a one-to-one mapping. We know MECP2 is the reason for this disease. So the correct interpretation of the methylation readout is really, really essential for neural operation after a certain developmental stage. And the interesting bit is that MECP2 has a very complex function in regulating gene expression. We also still don't quite understand how it is regulating gene expression. there's the initial assumption is that it's repressing, has a repressing function, but there was also evidence that there's activating function, that it has an activating function, and so it's much more complex, the precise mechanism of MECP2. What was also remarkable in Adrian's lab is they could show that these defects are in mice actually reversible. So they engineered mice with a defect in MECP2, but they were able to conditionally express MECP2, and in this case, the mice recovered and they could cure the disease in mice. And that gave rise to a huge amount of hope for people with this disease, affected people, that Rett syndrome is potentially curable. And indeed, this year, there have been the first trials for gene therapy for Rett syndrome, and I think it was last year that the first, last week or 2 weeks ago, that the first patient was actually treated. And so, in this case, earlier we studied MECP2 as a reader of DNA methylation, but also interesting are the writers and how this pattern is actually established and what function it has in the brain. So this is shown here. So what is interesting here is also that like the DNA sequence itself, because this signal, this additional methyl group is predominantly added to CPGs, so to see that it's occurring in the context of cheese. The reverse complement of that is also CG. So it's actually asymmetric. There's a symmetry involved in that. And this provides a mechanism for cellular heritability. So similar, like the DNA double strand, it can be, which is divided during cell divisions, such that one strand is going to both of the daughter cells. In the same way, the methylation signal can be brought forward to the next generation and then can be copied. And that is happening through a very intricate process. So we have these epigenetic writers. They are called, in this case, DNMTs. And there are certain different classes of DNMTs. So there's de novo methyltransferases, DNMT3A and 3B, which can establish de novo this methylation pattern. But then there's also a protein called DNMT1, which recognizes hemimethylated CPGs, so CPGs that have the methyl group only on one strand, and then they copy it onto the other strand. And that is, of course, very important. That is, of course, very important during cell division in order to propagate this information of where the methyl group has actually been added. So when the cells are then dividing, so the parental strand are divided into the two different daughter cells, and as a consequence, during synthesis, the newly synthesized strand would be unmethylated, and so you have passive demethylation during cell division. So these CPGs, which are methylated on both strands, will generate hemimethylated CPGs. If you have hemimethylated CPGs in the parental on this parental sequence, it creates unmethylated CPGs, of course. So every time the cell divides, the methylation is, first of all, basically reduced. So you have passive demethylation, which has to be reestablished through this DNMT1. But there's also active demethylation through this enzymes called TET enzymes. So you can actively, through certain intermediate stages, remove methylation marks to create unmethylated CPGs again. And this is really a very interesting system, I believe, because it does exactly what we have discussed in the beginning about epigenomic mechanisms. It provides a memory, and potentially a very long-term memory, such that events that might happen during childhood might be memorized for a very long time into the future. But it also provides an opportunity for dynamic changes, for example, for differentiation. And this is created through these kind of system of DNMT3A and Bs, which are able to write new methylation patterns, a protein that copies old patterns, DNMT1, and active and passive demethylation through passive demethylation during cell division, and active one with TET enzymes. And so this is very interesting, because if we are looking at the differentiation, for example, the context of the neural differentiation, it might provide a context where external stimuli can actually be integrated into a genetic mechanism, into the regulation of gene expression. And we were studying that in particular in the context of oligodendrocytes. So with Sabine, what we did is we were trying to understand what happens if we are actually getting rid of DNMT1 in the context of the neural differentiation. So we conditionally knocked out, well, she did, DNMT1 in a mouse model in condition of the expression of nestin, so for all the cells in the neural lineage. And this assumption was really, what is going to happen with that? Would that mean that the epigenomic memory, the cell identity maintenance, is actually kind of lost? Because that's what the function of DNMT1 is really good for. It's meant to maintain the methylation patterns over longer periods of time. So DNMT1, as I said, was deleted in the context of nestin Kreh lines, so affecting neural stem cells and all intermediate neural progenitor cells. And this is, and what you can see here actually is that, so this is the protein level of DNMT1, starting from embryonal. So at embryonal day 16.5, DNMT1 is actually very lowly expressed. But then at birth, in wild type, you have a very strong expression of DNMT1, which is slightly reduced over the following days. So our experiment affects DNMT1 from day 14, but it really becomes, the effects really become visible after birth or at birth. And we want to see how that affects the postnatal development. So there was an earlier experiment where they had created similar mouse lines, but these mice died at birth, so they couldn't actually study what happened in the first 2 weeks. So here we have a longer postnatal survival, and we can really compare, we can really see what happens in these mice in the brain development in the context of DNMT1 and methylation. And what is interesting is that, as previously shown, so the mice are burned without any phenotype, despite the fact that DNMT1 is knocked out from about embryonal day 14. So we have done quite a lot of experiments to really show that there's pretty much no difference at birth. But then in the next 2 weeks, the changes are very dramatic, and all of them are sadly dying after 14 days. And basically there's a complete growth arrest, developmental arrest. So they don't grow, and they have very severe phenotypes, which are also very similar to some of the phenotypes that we are seeing in rat models, mouse models. So as I said, the data set is not state of the art. So we have RNA-seq data from postnatal day 5, and so gene expression data and also methylation data from postnatal day 5. Sadly not single cell data, but whole brain data. But of course, in the last couple of years, there's a lot of data sets coming out that do study single cell expression patterns for the brain. And in particular, in 2018, there was a very good paper with looking at on single cell expression levels across the whole mouse brain. And what they did was they did it at P2 and P11. So right, and our data set is right in between of that. So that's quite handy. But it's really a huge challenge now if we can use that to deconvolute our bulk data sets, because here we have more than 70 different cell types alone in the brain, and it's a bucket of a very big mixture of cell types that are all to some extent affected by the experiment that we undertook. So my student Chris did quite a lot of work in improving deconvolution methods to really understand which cell types are affected to what extent, and to use the single cell data of the brain to deconvolute our bulk data. And this is a package that is going to come out very soon, but I'm not going to talk about it because it's not quite ready there. But we can now show that differentiate the 70 different cell types in our bulk data set. And we have been specifically looking at the differentiation of oligodendrocytes. And so this is the embeddings of the single cell data from P2 and P11, so the published data set. And you can see the data set starts from oligodendrocyte precursor cells. I'm sorry, I'm showing this slide again. So this is the differentiation maturation timeline pathway of oligodendrocytes, which go from the neural stem cells towards the mature oligodendrocytes. If we are looking at that in the context of single cells, at the two different time points, so at P2 and P11, we find that at 2 days after birth, the late cell types are not very common. So we have a lot of OPCs, precursor cells, but after 11 days, this has, can't see the cursor. So that's the green cells on the top. There's a lot more of these mature oligodendrocytes, which really differentiate in that crucial period after birth, in the 2 weeks after birth. So now we have created pseudobulk data sets and tried to deconvolute those. And we have then and validated our approaches there. And then we use our own data from the wild type mice, which are 5 days old. So they are right in the middle between the P2, 2 days, and P11. And we see that our deconvolution works in the sense that the proportion that we are finding for the different types of cells are quite in the middle between the two time points. And then if we are looking at the knockout cells where DNMT1 is missing, the methylation writer is missing, then we can identify the break point where the differentiation is not moving forward as a consequence of the missing methylation writer. So, this was quite an interesting, and I'm just giving you a very short introduction to this, but it was an interesting experiment. So we knocked out DNMT1 in the context of neural differentiation, and we were kind of with the idea that DNMT1 is really there to maintain the methylation profiles and the cellular identity, but what we find is that it's also really important for differentiation and for these dynamic changes that are occurring in this time span. So it really is important that you know where you're coming from in order to be able to make the next decisions, and we have a couple of ideas there as well. So just to summarize this project very quickly, what I wanted to tell you is how important DNA methylation really is in the context of neural development. It leads to a very pronounced postnatal phenotype if you are losing DNMT1, and we find, we could really, we were able to deconvolute a bulk data set to show that the oligodendrocyte lineage was severely affected by this loss. And the other things are not so important. So in this context. So we also looked at other questions there. Questions? No, that was just, I didn't show the method at all. So that was where we started from. We started from a out-of-the-box deconvolution method, but that was designed mainly for blood and for cancer, where you have maybe 5 or 10 different cell types, quite different ones. Here we have more than 70. Some of them are very different, and some of them, so you have the neuron lineage, you have the clear lineage, you have oligodendrocytes, so you have to have a little bit different approaches to get the different. It is not, so the method is not published yet, that's right. So we are just about, I think it will be on bioRxiv in probably 2 weeks, and then I would talk about that a little bit more, but at the moment not yet. And also I wanted to give you some ideas of some other projects. Right. So we also looked at DNMT1 in tumorigenesis. So the take-home message from the, is there a question? I mean, we cannot describe, so what we did here is we used the bulk data set so one of the earlier experiments that we did, the bulk contains more than 70 cell types, and we deconvoluted it with a subset of the single cell data, which only contained the oligodendrocyte lineage. And so we can, despite the fact that in the bulk data set we have more cells, we are able to correctly identify the relative proportions of the different oligodendrocyte cell types. So that was one initial experiment, just focusing on this small subset of these 10 groups. But of course, what we cannot do, well, what we haven't validated so much is predicting completely how many completely so reference-free deconvolution. I think in this context with so many cell types is very difficult and also difficult to validate. But I mean, if you're focusing on a subset of cells and trying to find the relative proportions of that is working well. That you could identify completely new cells is much more difficult. It depends on, the mixture, I think, to a large extent. So I can't say that in general. Okay. So again, from the biology, so this was neurodevelopment. So on the other hand, I also wanted to very briefly mention, just from the biological findings, that methylation is also incredibly important when you are studying cancer tumorigenesis. So in this case, we have again looked at DNMT1, and we have a similar model of our DNMT1 knockout, in this case in tumorigenesis, in this collaboration with Gerda Ecker, who's also in Vienna, and her student Elisa Riedel. And in this case, we were looking at a tumor type called NMPM-ALK positive anaplastic large cell lymphoma, which is a very rare and very aggressive non-Hodgkin's lymphoma of T-cell origin. And it's driven by a constitutive activation of the oncogenic anaplastic lymphoma kinase ALK. So if you have this NPM-ALK fusion gene, then this will deterministically lead to T-cell lymphoma, and we have mouse models again for this kind of problem. So these NPM-ALK transgenic mice will develop tumors in the tumors, and they will die at the age of 15 to 30 weeks. However, if we are looking at double mutants where DNMT1 is knocked out, this reprogramming into a tumor cannot happen. So we have 4 different models. So we have the control tumors, we have a DNMT1 knockout again, then we have a tumor model where you can see on the right side that it does develop this big massive tumors, ALK, and then we have the double mutant transgenic mice which do have the tumor gene, but they also don't have DNMT1. And in these cases, the tumor doesn't develop and it prevents the reprogramming. And again, we had to use quite a lot of deconvolution tools to analyze this data because it was our bulk data sets. But what that tells you here is that, so it's very well known that there are methylation changes are hallmarks of cancer, and they are really important to drive massive reprogramming changes. So you have, when you look at DNA methylation, you have a very characteristic landscape where you have around promoters, you have unmethylated regions, and in the bulk of the regions, bulk of the genome, you have methylated genomes, which is basically repressing. And in cancer, these kind of patterns are disturbed. So the CpG islands, which are unmethylated, become hypermethylated, and the bulk genome becomes hypomethylated. And this causes a lot of reprogramming. So again, just if you're interested in tumorigenesis, DNA methylation is definitely something that you want to look at. With that, I would want to move track completely. I'm talking about a third project. How much time do I have? Oh, okay. Half an hour without questions. Okay, so the next thing is that's a little bit more machine learning. What we wanted to do is we wanted to do epigenomic imputations, and again, we developed a new tool called EDICE. And this is work with my students. Mainly it was started by Alex in the top corner and then taken over by Giovanni Tamaggi, and Chris also helped, and I'm mentioning in the end as well. So histone modifications, I've already mentioned them. So we have these nucleosomes which create this pearl on a string structure for the DNA sequence, and then the yellow, somehow my cursor is completely gone, but these yellow bits are the nucleosomes, and you have the yellow circles and the red triangles, which should symbolize that the histone proteins again can be modified in a similar way as the DNA sequence itself. And that looks like that. So you have again the histones, which are the yellow balls where the DNA sequence is wrapped around it. If you look at the histones, that's one of them is this kind of octopus on the left side. So you have an octamer of 4 different proteins in the middle: H2, H2A and B, H4 and H3, and they have long arms, and you see the amino acids along these tails. And if you're going, for example, on protein, the red protein H3, and you're going onto the tail position 4, you reach a lysine K, and that gives you the position H3K4. And this position specifically can be methylated, mono D or trimethylated. So you have, for example, histone modification called H3K4 trimethylation. But you can see there are these 4 different tails, but in fact it has 8, so it's a dimer. And you see the different positions along the tails, and every one of these little circles is a modification. So you have a very complex patterning of these histone proteins. And again, these patterning can be measured. We can measure them in a sequence-specific way, so very localized on the DNA sequence itself. So for every single position, we can have a measure if these histone proteins are modified in a given way or not. Does that make sense? Or did I lose everybody at that stage? Okay, so let's try. So this is again the genome view. So you have your long DNA sequence, so your long string in blue. You have the yellow histones, and you have, for example, in this case, we are focusing on one histone modification, H3K4 methylation. In the human body, because it never is easy, you have actually 6 different writers. They are called KMTs. They all write this single modifications along your genome. You also have erasers, so proteins again, which can remove the mark. And so chariot 1A is one, for example, and you also have epigenetic readers that are having domains that very specifically recognize these particular histone modifications. And then we can measure these histone modifications across the genome. So that's kind of shown here on the left. That's a very small part of the genome. Again, it's actually from a yeast genome. So the x-axis is the genomic axis. You see on the top a couple of genes, which are, the green one is expressed, the red one is repressed. And the different tracks are now different histone modifications. So one of them is H3K4 methylation. So you can see now how it affects the whole genome, and you have the different histone modifications which also give rise to a track like that. And as I said, there's a strong correlation between individual histone modification and gene expression. So for example, we know that the mark H3K4 methylation is very often occurring at the start of genes, and particularly at the start of active genes, while H3K27 is usually found close to repressed genes. And that gave rise to the idea that there's really a histone code, that histone modifications can actually control gene expression. Very, very difficult still to understand what they actually do. It's not very simple. But you have readers and erasers and writers, and you have a correlation to a readout, which is the gene expression. So is it an instruction for gene expression? But we also know that there are dynamic changes. So histone modifications change over differentiation patterns and during development. So that means that in every single cell type, you have different histone modification patterns. Again, when we look at neurodevelopmental diseases, so if you're just looking at this one family of proteins, the KMT proteins, which are the writers for H3K4 methylation, the 6 different writers, they are all involved in so in some cases there's one-to-one mappings again between the individual, the gene and the disease. So they are quite causal there's associations, but also causal association between mutation in this gene and neurodevelopmental disorders. And also, they are very important, seem to be important in a different number of cancer types. So it's really important to understand what these histone modifications are there. They are not redundant, these 6 different writers. So many of them are embryonal lethal if you're knocking them out. So of course that comes with a lot of challenges now. So we have these data sets which are very high dimensional. So we have maybe 100 different tracks or more. And they are cell type specific. So if you are imagining analyzing genomes is difficult, epigenomics analysis in this context is much more complex data sets. So it's high dimensional, and as you also see, the tracks are not independent. They are kind of interacting in complex ways. And also the data, I mean, we have large data sets, but the data is still quite sparse. Because each cell has a different epigenome, and the assets are still quite important, quite expensive. So that gave rise to the imputation challenge, basically to fill in missing data points. So in this data matrix, you have on the y-axis the different cell types, and on the x-axis different histone modifications. And the blue points are the ones where we have measurements, and the white ones is where we don't have the lack measurements. And the idea is, could we come up with deep learning, with machine learning methods to impute missing data sets. So there was a challenge by the ENCODE consortium to do exactly that. So to predict histone modifications in cells where they haven't been found. And we took part in this challenge, and here's the data matrix that we were given, again, very similar. You have the histone modifications on the y-axis and different cell types on the x-axis. The blue points are training data sets, orange is validation, and green is testing test sets. And it's important to note that for this challenge, the green data sets were specifically generated, so they were given us after the initial phase of the challenge to make predictions on them. Or they were actually not given to us. They used them to validate our predictions. And most of the data sets, the blue ones that we had for training, were gathered in this database over a long period of time. So they were also experimental developed. So the experimental design, the technical things also changed between the training and test set. So which made that the validation also very difficult. So we, yes? Sorry? So we have some histone marks. So for example, H3K4 methylation is a very well studied modification. So we have a lot of samples for it, as you can see on the right. So it's the top histone modification. But we also have histone modifications like H3K9 demethylation at the bottom, where there's only one sample. Okay, so there's a wide range of, and the same holds for cell types. So some cell types are very well studied, and others are very well understudied. On the x-axis are the cell types, and on the y-axis are histone modifications. Yeah, that's a very good question. So that was the challenge of the challenge design, I guess. And so I think initially they used a lot of cross-validation ideas that and they looked at how imputation methods worked on well-studied cells and histone modifications, and then they found that when you actually apply them to a new cell type or a different histone modification, they don't work as well. So the idea was really to also understand other cases where you don't, but it made the validation really, really complex. And I'm going to talk, if I have time, a little bit about that. So the competition ran in 2019. The paper came out 2023. We had a prototype with which we entered the competition. It was called IMP, and we did actually quite well. So we got third place in that case. So I'm going to talk a little bit about why this is working and what we did. So why is it working? Well, the idea is why does imputation work, or why can we hope that it works? Well, we see that there's partial correlation between data sets, and this partial correlation stems from the fact, one, that histone modifications are not independent of each other. That means that epigenetic writers actually contain reader domains. So and you can kind of learn this relationship. But I've already said, like for H3K4 methylation, there are 6 different writers, but it's just one track. And these writers have different reader domains, so they interact with different other histone modifications. So it's really complex. But we know that in a given cell type, there's a correlation between different histone modifications. And then also, histone modification are partially correlated across cell types, so because they are also marking like functional elements, like for example the start of the gene or gene bodies, and these are of course encoded in the DNA sequence, which is the same across all different cell types. So there's a partial correlation across cell types as well. So the idea would be you're given these blue data sets from the blue circles and you want to predict a new combination of histone assay and cell type. And there were a couple of earlier methods, in particular Chrome-Impute, which used an ensemble of regression trees. So the disadvantage of that was that you had to actually retrain a new ensemble of regression trees every time you had a new target cell type assay combination. So it's very accurate. It looks directly on the data, but it's not very flexible at all. And then there was work by Jakob Schreiber predicted an avocado, and they formulated it as a tensor problem. So with one dimension being the cell type, one the assays, and one the genomic positions, and then they had different ways of combining the different dimensions. And so in the last, in avocado, they used a perceptron for the final prediction. But this is really it's a very big model, and it can't be trained on the whole chromosome, basically on the whole genome, because of the level of the, so basically as an input, you would just take the index into the tensor and then make a prediction. Well, there's a lot of redundancy in the genome. So your for example, intergenetic regions look very similar. So they are not actually using the data set, the measured data themselves at a given genomic position, but they are using it as a factor. And that's computationally not very efficient. So we came up with EDICE, which is epigenomic data imputation through contextualized embeddings, and is kind of taking the best of both worlds. Again, we are starting with this kind of tensor. So in this case, but every slice of the genome is treated as an independent sample, and the task then is to predict, so given given the measurements in that genomic bin for a number of cell types and a number of assays to predict the missing values. So we are, sorry? No, it's a continuous value. So we use signal embedders. We combine the local embeddings with a global cell type embedding that we are learning. And this captures the information of each genomic bin. And it combines the local and the global genomic embeddings. And then we are using a self-attention block to contextualize over the different cell types. And we are doing the same type. So this is the self-attention, includes the interactions across the cell types. And then we are doing the same thing with the assays. So we are again looking at have a global and local assay embedding, and then contextualizing that with a self-attention model, and then we are feeding that into a multi-layer perceptron to come up with the final prediction. So the advantage of that is that it generalizes really well to unseen assay cell type combinations without the need for retraining. So we don't need any genomic embeddings, and that means that the model size is really scaled down massively. And it also means that the architecture is really suitable for domain adaptation if we get new data sets. And that has allowed us eventually to create a model that we can use to predict individual, so personalized tissue-specific histone modification patterns, which is quite novel. So in this competition, we had to predict a reference epigenome. We had to predict reference epigenomes. So that means that the tissues were actually not coming from one individual, but it was just a reference epigenome coming from different labs, really, where there was it wasn't individualized as in individual persons. So just to say, so we are having a single model, and we reduce the numbers of parameters per model by a number, several orders of magnitude. We can also now train on a much smaller data set, to be honest. So on here what you're seeing is the number of training bins for genome-wide prediction on the x-axis, and the genome-wide correlation between measurement and correlation on the y-axis. And for avocado, you need to train on the whole thing because you have the genome because you have the genome factors, really. But we can achieve better accuracy already with a much smaller training set. And this is just a zoom in one of the tracks. So in blue you see the measurement, and in red you see our imputation, and it works really nicely. And on the bottom you see what we call a peak. So that's called with a tool called MAX, and so the measurement and the imputation work quite nicely. And then we have all kinds of different ways of comparing imputation and measurement, which is really took us the last couple of years, and also the competition organizers have been struggling for a couple of years finding out how to really compare measurements and imputations. But for these kind of statistics, so EDICE is the blue one, and you see that it's usually, so for example, mean squared error in the top left corner, the blue one, EDICE, does really well on these things. But the problem is, when we validate these predictions, is that we are actually wanting, and that is really important, I think. What we want to do is we want to find the needle in the haystack, okay? So we might be able to completely copy the image, copy the histone modifications correctly, and they can be very similar for different cell types, for example. But there might be one needle that is different between the two different cell types, and that is the one that we want to predict. And correctly, because if we can't correct that difference correctly between, for example, tissues, then it's to some extent useless, because we are interested in the differences. But the problem is also that there's not really a gold standard for telling you where the needle is in the measurement. So the validation, if we are just comparing, the measurement with the prediction, and we miss the needle, then we might get very high scores overall in the validation, but it's biologically not relevant. It doesn't tell us what we need. So part of it is, so histone modifications are only enriched in small parts of the genome, some of them, so we have to find these small parts of the genome and make the right predictions, but also the differences between tissues might be very small. So, and we want to see specifically if we are able to predict these differences correctly. So in the top you see a peak that is the same in two different cell types. So the cell type is here E25 and E52, and so you have in blue is E25 and in red is E52, and then you have the imputation and the prediction, and the top peak, okay. I have half a talk still to give. I'm not stressed, I don't know. Okay, but what I'm trying to say here is that we are actually able to make very good predictions also of peaks that are tissue specific. We try to validate that with the Wasserstein distance to quantify the difference between peaks in different cell types. And so the Wasserstein distance on the y-axis is the observed one between two tissues, and on the x-axis is the Wasserstein distance between our imputation, and that correlates very well, and you can see which peaks I have chosen for these two examples. So we are actually able with our tool to predict very well tissue-specific differences in the histone modifications. So it's not just that overall, the peaks are similar, but differences are picked up nicely as well. So I want to say this as actually it's very important for me, so despite that I'm now at the end of the course. But I think you have to be really, really cautious when you do machine learning, even big models, and you make predictions. What are you actually doing there? How do you validate it? So in June 2020, at the initial round, so that was done in Stanford, they validated all the different imputations, and they came up with a ranking of the methods. And then what was really striking for them is that as a baseline, they used just an average over the histone modifications over all tissues, for example, all cell types. And the average model actually outperformed all of these other very, very complex algorithms. But then they thought, no, that's maybe it can't be true. And they changed something in their validation and in the way they looked at the data. And they came up with a completely different validation. So that was 2 years later. And you can see that average is now at a different rank. So sorry to explain that. So these are on the x-axis are the different tools and methods that have been participating in the competition. And then on the y-axis, the average score. So bottom is good and top is bad. And now the ordering is completely changed after 2 years. We are still doing reasonably well, but the ordering is completely changed. And in between, in June 2021, there was yet another ranking where the ranking was changed again. And I have to say, this is the same method, same predictions compared against the same measurements. So they have changed to some extent the way they are ranking them the same, and also what they have done is they looked at the data set and they found that between training and testing there was what they called a distributional shift, which is technical. So the technical circumstances changed and the pre-processing changed as well. And so now they corrected for that. They corrected the test set. Pre-processed them in the way the training set had been processed, which didn't really match with the experimental procedure that they had used for the testing. And they also introduced quantile normalization. And that led to the fact that these peaks were, so the shape of the peaks were changed dramatically. So this is the experimental data after pre-processing. This is from the training set. This is the initial test set. Then after quantile normalization, they came up with this set. Then they changed the pre-processing, which led to this peak. And then they combined the change of the pre-processing with the quantile normalization. And then they came up with this signal. So, and the shape changes, the height changes, the dynamic range changes. And therefore, the validation of the method changes completely. So to me, this was quite striking that you have to question what are your measurement? Can you trust your measurement? What is the gold standard? If you compare across tissues, what do you pick up and so on? When you compare data sets from different labs, it's quite big, to be honest. I'm not sure I can, so we thought about that for quite some time to try to find a way to get the systematic error out of the data set by actually combining multiple histone modification from the same experiment, saying that they would share the systematic errors and that we can in a more principled way, pre-process these data sets. And we looked at the experimental design for that. We know that there's the antibodies that are used to detect the histone modifications are not 100% specific. So we have some unspecific binding as well. Usually you have a control sample, what we call a background sample, where you don't use the antibody against your histone modification. So the red thing here again is the histone modification. You do the treatment sample, with using an antibody that recognizes these histone modifications, but it's not 100% specific, and this unspecificity varies between assays. And then you do the same thing, but without the antibody, because your sequencing introduces a lot of biases depending on the DNA sequence itself, whether it's compacted and so on. And so you have both the mixture of specific and specific and non-specific reads, which we removed by using, again, non-negative matrix factorization, and then also and then you also have the systematic problems with the sequencing and alignment and so on, and we solved that by using half-sibling regression to correct for the signal. So this is the convolution of the non-specific versus the specific bit to make the data a lot cleaner across different histone modifications, and then and it gives us the mixing factor that is proportional to the specificity of the different histone modifications. And then we remove the multiplicative factors, which include accessibility and mappability, and we are getting, and that is done by also with an actually causal model, half-sibling regression, to predict the hidden underlying true modification patterns. And that's what we've been doing here. And what you can see here, this correction works quite nicely. Before we have done that, we see that if we are comparing the background, the control samples with the signal samples with the different histone modifications, you see that there's a large number of bins which are highly correlated. And this is particularly true for some of the more difficult histone modifications like H3K27, so where the control sample is very highly correlated with the actual signal. And we can remove that and then get the technical biases out to a large extent. So I think that's a word of caution. So what is the gold standard if you do imputation? What is ground truth? You really have to understand the experimental procedures and the computation pre-processing. Don't just use a data matrix, and then put it in a black box machine learning and have some predictions. Usually that is not very informative. You have to understand both the experimental procedure and the biology behind it, I think. However, we are quite positive now that we can do tissue-specific histone modification, imputation, but the validation is quite difficult. And we now went on to do personalized epigenomic imputations. So in this case, we get data from deceased donors. So that's really their data sets, which have been published rather recently. So there are 4 individuals, males and females, and we get over a large number. It's still the same data matrix. So we have different tissues, different histone modifications, and then the different color codings tells us now the individuals from which we get the data set. And again, we can do individual, so we use transfer learning in this case to make individual specific predictions. But many of the of the enriched regions are actually shared between individuals. You see that here as well. So you have the 4 different individuals. And however, we do also find particularities between individuals. We can call those epi-alleles. And we are also relatively good in predicting those correctly. And it's, of course, again, more complex because now you have the dimensionality of the different individuals in the different tissues. And some of these changes, these enrichments are particularly subtle. So you want to have a very good method there, and you want to be confident as well. And this just shows you that some of the histone modifications are really shared between a lot of histone modifications some of the histone modifications are shared between a lot of individuals, or all of the individuals and a lot of tissues. And some are very unique for a tissue and for individuals. And those are very difficult to predict. And I think I'll just leave it there. Yeah.
Okay, questions?
Thanks for a great talk. I've got lots of questions that I could ask, but I'm going to limit myself to one. So CHIP, there are a bunch of new assays that measure histone modifications and transcription factors genome-wide. So I'm kind of thinking of cut and tag, cut and run, those sorts of things, which are generally a little bit cleaner than ChIP-seq, but I guess they're still going to have their sources of noise. So have you kind of considered adapting these sorts of models that you've been using to analysing the kind of newer data that's coming out?
Yeah, I think that's one of, so we are using some of this data set to validate the older ones, or the predictions for that. I think we still need a large, in general, we like to work with a larger number of tissues and histone modifications, and with the newer ones, this is only building up. And for the individual, so the personalized epigenomes, I haven't seen a similar data set yet. So like this one came out, I think, in 2022.
It's closer to you.
Hi, it's a great talk. So I think when we deal with genomic data, we always have to consider ethnicity, right? When we talk about epigenomic data, do we also have to consider different ethnicity? Like, for example, you take like the epigenetic data from 4 samples, right? Are they from the same ethnicity, or do you think we should consider that? Thank you.
I think this is an amazing question, which brings me to my last point actually in the talk, which is that I think ethnicity is just one thing that we probably need to consider, and that we don't quite know how yet. But I think if it's important for genomics, it's even more important for epigenomics. I think the amount of information that is encoded in the DNA is really massive, and we understand that now, and it becomes available over a large number for a lot of people, and I think it's really important to consider that. So we know that in the genome there's so much information about ourselves. It tells us so much about us. I mean, not just, these protected categories like sex and ethnicity, but and wellness things and funny things and useful things. So genetic health risk reports, for example, it tells stuff about us, but it also tells about the past, of course, our ethnicities. It also tells about the future, if I'm a carrier of something. So it's not just, this information is not just about us, it's about the people that are like us in whatever aspect, but also that may come after us, and we have a responsibility to consider that, I think. It tells us a lot of things. So there's a lot of things that are not in my DNA, so all my background, but that is important for me as a researcher, and I think that is really, really important that we are approaching these questions as so we are our own study objects, human. So it is a lot about who we are in the world and how we do that. So where we come from is really important, and we have to understand that in order to do better science. So we have to learn who we are and understand our biases. And I think for machine learning people, that's still sometimes difficult. And for me, it was difficult to understand that we are not just doing objective science, but we are approaching our questions with this background. So the questions we ask as scientists and the answers we find are shaped by who we are and the role we play in the world. what we teach is to the best of our knowledge based on well-established facts and reproducible science, but how I present it to you today, how I did that, really depends on the selection that I make, the ordering, the weights I give to different points. It's determined by my own framework. And you see scientific facts through the lenses of my filters. And epigenomics is really fascinating because it taps into that. There's a big assumption that it doesn't. So with epigenomic data, we always get the DNA sequence for free. So we always get all this information for free. But we get a lot more as well, potentially. So because there's a lot of interaction there between the environment and what happened early in our history that is stored in our epigenome. So, for example, I'm now involved in a project where we are trying to understand how early childhood events have an effect on how we experience pain later in life and how that affects chronic pain and also drug abuse. And there's good evidence that methylation is or epigenomic mechanisms is a mediator that stores this information of what happened early in our life and preserves that throughout our lifetime. So it's very well, so we can only speculate because we don't have these large data sets yet, but that, for example, socioeconomical situations are also ingrained to some extent on our epigenome. And that raises massive questions. And of course, and it's not the data set alone. So just by selecting more and more data, we cannot hope to be more objective. Because the very act of measuring and collecting data, of course, involves interpretation and value judgments. And so, I mean, this is from Judea Pearl's book, the Book of Why, which is about causality. And it really says that data alone is hardly a science, regardless how big they get and how skillfully they are manipulated. So it's really important that we understand that, first of all, data is not objective, and we as researchers are also not objective, in particular when it comes to us studying ourselves. So there's massive implications coming with epigenomic research and also with machine learning being used to study these kind of data sets as they become available. So, for example, social and political structures could influence the risk of epigenetic-based diseases. So socioeconomic classes are the most epigenetic disfavored ones. But it could also create social discrimination and inequality to the universal access to epigenetic-based medical care. We could discuss on how evaluation of the consequences of lifestyle habits have influences on the health through epigenomic markers, and how that the limit of individual responsibilities for their own or even the next generation of health. So I think there's a huge conversation that we have to have, which is not just the machine learning people or the computational people and the epigenomics people. We cannot have these conversations on our own. We have to widen this up to a much bigger conversation, because the implications are beyond what science can do, I think. And for that reason, we have created a new group in Dundee where we get people from philosophy, political sciences, also law and ethics to come into this conversation to understand better what the implications are, because many we don't quite understand yet. So I don't think that we can do that in a two-way conversation between statistics or machine learning and medicine or genomics, but we need a bigger conversation about that. So really my take-home message at the end of that is, as we move this machine learning into health and biomedical data analysis, we have to be very much aware of our own biases. So for me personally, when I started working on genomics, I started with plants. And then I moved on to study worms. And then I started to look into mouse. And only very recently I started to work on human data. And data really there's no difference for me. I get the data and it's on my computer screen. And when I showed you the data with the brain data, for example, it didn't make a difference to me that there were mice sacrificed for these data sets. And sometimes I would say, look, I need more data. We don't have enough power. And my collaborator would go back to the lab and she would sacrifice mice. But I'm completely removed from that side of the research. It doesn't make any difference. But now as we move into, or as I moved into working really with this health data and with personalized data, I think we have to understand much more about the implications and also learn a lot more and be more open about what are actually the ethical considerations that we have to consider, and how do we design experiments, how do we make sure that it's fair.
I'm glad you asked that question, because she had a whole rest of the talk about it. I think we got time for one more before coffee.
I can also say thank you to my team now again.
Yeah, I'm interested to know, of course you haven't measured it, but I'm interested in your perspective, because what I'm seeing, I work with genomics data, and currently the breakdown of diversity in terms of ancestries that you find in major data resources are somewhere between 80 to 90%, depending on the data set, European. And then you have like 1% African, 1% indigenous, and so on. So there's a lot of bias. Then we also found in our research that there's also quite a lot of sex bias, in the sense that, and not just because it's a consequence of, deliberately trying to do this kind of bias. For instance, you don't have, it's very difficult to have pregnant women for studies, simply because of ethical concerns. So I'm very much interested in understanding the current biases in the global data set landscape, and so I wanted to know your opinion on where we are at when we talk about the data sets that you've been working with so far.
I don't have answers, proper answers, but I think we have to start with the ethical questions and the implications at the very beginning of any experimental design, rather than what it very often happens that we do it at the end. I want to say that I think the bias in the data in the data sets is something that is very often mentioned, but I think there's also biases in outcome. So, for example, there was this 2020 science paper that showed that algorithms that are used actually already in quite far usage to manage the health of populations in the US. They are optimized for costs, really, where the prediction, where the cost of post-treatment, for example, is used as a proxy for success. And that means that if you have groups that are coming potentially with many comorbidities, in this case, the follow-up costs will always be higher. And if you're using the costs as a proxy of the success of the intervention, for example, it can lead to the fact that you are excluding people from getting access to treatment which are already more vulnerable. And so I think it's not just the bias in the data, but it's also your optimization function. So, and what do you use to optimize your function? That can have massive, and that's less well studied and understood. And very often these cost functions are also opaque if it's getting more and more into also the private sector. So the I think transparency is already one good thing. The other thing, I was talking to someone at GlaxoSmithKline, and they said, well, you have to think much more if you tech biases in your training data sets, as you say. You have to come up with new strategies to actually start going out in the world and generating more data sets. Something also from a purely machine learning point of view we might not want to do initially. We just deal with the data that we got, but you have to have strategies to collect more data sets, I think. I don't know if that's a complete answer.
Thanks. Let's thank Gabriele once more.